Welcome, everyone. It's fantastic to have such a wonderfully diverse and specialized group here today. Whether you're a food scientist formulating the next big plant-based meat, an analytical chemist isolating complex molecular structures, a toxicologist focused on consumer safety, or a regulatory pro navigating those tricky global frameworks, well, this explainer is exactly for you. Today, we're moving way beyond just the surface aesthetics of what we eat. We're going to examine the strict molecular reality behind everyday food colors, drawing exclusively on rigorous structural chemistry and biological data. So let's dive right into this fascinating molecular world, shall we?
Let's kick things off with a fundamental physical chemistry problem, especially for the analytical chemists and food technologists tuning in. Picture a food matrix. You extract this vibrant, intense red pigment at a low pH. But then, when you formulate it into a slightly alkaline product, it suddenly turns deep blue. Why does this happen? The answer isn't magic. It's all about structural transformation.
And this brilliant structural layout illustrates the exact mechanism. What we're looking at here is anthocyanin, a flavonoid glycoside. You can think of it as nature's molecular transformer. At a pH of 1 and below, it exists as the flavylium cation, which gives us that intense red color we naturally associate with apples or strawberries. However, as the pH increases to between 7 and 8, a structural transformation actually occurs, yielding the quinidal base, which presents as a deep blue. Push the pH even higher, and it shifts to green and yellow. For our formulation scientists, understanding this precise structural equilibrium is exactly why anthocyanins are mostly restricted to low pH foods, like fruit syrups and soft drinks. Here is a quick look at our agenda. We're going to cover 5 key areas today. We'll start with nature's molecular color palette, then explore some unexpected biological sources, and after that, we'll dive into color stability science. We'll wrap things up with analytics and regulation, followed by a look at the biotech future of pigments.
All right, Section 1, Nature's Molecular Color Palette. Let's look at nature's foundations.
We're stepping right into the vast plant kingdom now. Take a look at the architectural consistency of the carotenoids. Nature has actually isolated over 500 of these. Look closely at these structures: lycopene, beta-carotene, capsanthin. For the organic chemists among us, you'll immediately recognize that C40 hydrocarbon chain, made up of 8 isoprenoid units. Notice the alternating double and single bonds? A minimum of 7 conjugated double bonds in this tetraterpenoid molecule is required before the compound even has a perceivable color. It's helpful to think of these conjugated double bonds as precise light-catching antennas. The extent of these bonds, along with the molecule's stereochemistry, determines exactly which wavelengths of light are absorbed, and that's what produces the brilliant yellows and oranges we see in things like carrots, tomatoes, and palm oil seeds. Now let me share a fascinating little fact that perfectly bridges agricultural science and food economics. 140,000. That is the exact number of stigmas from the Crocus sativus flower needed to produce just 1 single kilogram of saffron powder. Saffron contains crocin, which is a water-soluble diagentubeoside ester. Because of this massive agricultural requirement, saffron costs around $1,000 per kilogram. And that incredible cost really dictates its application. Food technologists use it extremely sparingly. It acts not just as a colorant, but as a very specialized spice to deliver its characteristic flavor through compounds like picrocrocin and saffronol. Moving from the yellows of saffron to the greens of leaves, we arrive at chlorophyll. Here we see the classic porphyrin structure: 4 pyrrole rings joined via methane linkages. And sitting right in the center is a magnesium atom held firmly in place by 2 covalent and 2 coordinate bonds. But for our formulation scientists, the really crucial feature is stretching out on the side, the phytyl side chain. This 20-carbon monounsaturated alcohol acts as a long hydrophobic tail. It's this specific side chain that dictates chlorophyll's insolubility in water. If you hydrolyze it and remove that phytyl chain, you yield chlorophyllide, which instantly increases its solubility in polar solvents.
Moving on to Section 2, Unexpected Biological Color Sources. Let's talk about microbes and insects.
We're stepping away from terrestrial plants now and diving straight into the microbial kingdom. On one side, we have pigments produced by the fungus Monascus purpureus. Historically, these have been used to color Asian red rice wines and salted meats. Natively, these Monascus pigments, like Monosca rubrin, are orange, yellow, and completely insoluble, bound tightly to the fungal mycelial membrane. To get a water-soluble red for food applications, biotechnologists actually have to react these extracted pigments with water-soluble proteins. This clever step substitutes an oxygen atom with a nitrogen from an amino group. Contrast that with what we have on the other side: phycocyanins,
from the blue-green algae spirulina. These billoproteins are naturally water-soluble. They're formed by 4 open-chain pyrrole rings bound to an aproprotein. Japanese companies have already successfully commercialized this vivid blue for use in soft drinks and fermented milks, having proven its safety through rigorous chronic toxicity studies.
For the toxicologists and nutritionists reviewing safety and sourcing, we absolutely have to discuss this structure, carminic acid. This is an anthocyanone, part of the quinoid pigments, and its biological source is highly unique. It's painstakingly extracted from the dried bodies of the female Dactylopius coccus costa insect. This insect actually lives as a parasite on cactus plants native to Central and South America. The insects are harvested, dried, and then treated with hot water, or more recently proteolytic enzymes, to yield this specific compound.
The really crucial point here is how we utilize that carminic acid in industry. On its own, at a neutral pH of 7, carminic acid actually has very little intrinsic color. To make it useful, we rely heavily on coordination chemistry. By complexing carminic acid with metals, most typically aluminum, we create carmine. This physical chemistry transformation gives us a highly stable, bright red colorant. It's virtually ubiquitous in commercial jams, syrups, and baked goods. And by precisely varying the ratio of carminic acid to aluminum, a chemist can dial in very specific shades, ranging anywhere from a pale strawberry to a dark blackcurrant. Remaining in the animal kingdom for a moment, let's examine heme. Compare this structure directly to the chlorophyll molecule we saw just a bit earlier. Just like chlorophyll, heme is built on 4 pyrrole rings. However, the central atom is no longer magnesium, it's an iron atom. In living animals, heme acts as an oxygen carrier in myoglobin and hemoglobin. When the iron is oxidized, it's a bright, vibrant red. But when heated, that loosely bound oxygen atom is lost, and that results in the characteristic brown color we associate with cooked meat. This is a genuinely critical structural insight for food technologists who are currently developing plant-based meat analogs and want those authentic color transitions during cooking. Let's shift gears to Section 3, the science of color stability and how we face processing realities.
Now that we've established our molecular palette, we have to confront the harsh realities of food processing. Let's detail some specific degradation pathways. Anthocyanins, while absolutely beautiful, are incredibly fragile. They are actively decolorized by ascorbic acid, oxygen, and sulfur dioxide. For carotenoids, their greatest strength is also their biggest weakness. Remember those conjugated double bonds, the light-catching antennas? Well, in the presence of light, metals, or lipid hydroperoxides, those numerous double bonds suffer what we can essentially think of as oxidative short circuits. That leads to trans-cis isomerization and, ultimately, color loss. Meanwhile, betanin, the red pigment extracted from beets, is highly sensitive to light, heat, and air. Because of this, it's strictly relegated to short shelf-life items or low-heat dairy products like yogurt. Interestingly, in those dairy applications, the food's high protein content actually provides a protective matrix against degradation. So how does the industry fight back against these stability issues? Let's walk through this industrial mitigation strategy for chlorophyll. Natural chlorophyll degrades very easily under process realities like pH shifts and heat. To solve this, manufacturers extract it and perform an acid-catalyzed hydrolysis. This purposefully removes the central magnesium atom to form a compound called phaophytin. Then they complex that phaophytin with copper. By replacing the magnesium with copper, they synthesize a highly stable commercial colorant. It's made available in both oil-soluble and water-soluble forms, boasting vastly superior resistance to both heat and light. Let's move on to Section 4, Analytics and Global Regulation. We're zooming out from the molecular level to the macro level now. Assaying natural colorants is incredibly complex because in reality, you're rarely dealing with a single pure molecule. You're almost always dealing with a mixture of pigments and their derivatives. Now, spectrophotometric analysis offers fundamental baseline scanning by measuring light absorption at maximum wavelengths. However, for true precision, analytical chemists rely heavily on high-performance liquid chromatography, or HPLC. When coupled with a photodiode array detector, HPLC gives us immense high-resolution power. It can cleanly separate and detect individual pigment components in complex matrices, like isolating specific anthocyanins in a cranberry extract, for example. From analytics, we naturally move to the global regulatory landscape. The legal classification of these molecules varies quite significantly around the world. In the United States, the FDA classifies natural food colorants as uncertified colors. That means they are exempt from certification provided they have GRAS status, generally recognized as safe. This includes items like annatto and beet powder. Conversely, the European Economic Community's Scientific Committee for Food utilizes a different framework entirely, based on acceptable daily intake, or ADI groups. It's absolutely critical for regulatory scientists to note that the EEC approves certain natural pigments, such as curcumin and lycopene, which have completely different regulatory pathways globally. Fortunately, efforts by the joint FAO/WHO Codex Alimentarius Commission are actively working toward harmonizing these complex international guidelines.
And finally, Section 5, the biotech future of pigments. Let's look at next-gen sourcing.
For the pharmaceutical scientists and biotechnologists joining us, this is where your expertise really shines. The food industry currently faces massive bottlenecks in natural pigment production due to low agricultural yields, seasonal variations, and unpredictable weather. But we can trace a really clear evolutionary timeline in pigment sourcing. Centuries ago, we relied entirely on agricultural whole plant extraction, which is highly vulnerable to climate and soil variations. By the 1980s, we saw a major shift. Researchers discovered that fungi like meniscus could be cultivated in controlled, submerged liquid cultures, making them completely independent of the weather. Today, biotechnology is allowing us to move away from traditional agriculture entirely. We're utilizing highly controlled advanced bioreactors and sophisticated plant tissue cultures to produce secondary plant metabolites with absolute predictability. To really illustrate the power of this shift, take a look at this number, 845. This represents a stunning biotechnology victory by the Japanese company Mitsui Petrochemical Ltd. By utilizing optimized plant tissue culture cells, they are actually able to produce 845 times more of the red dye chiconine than what could be traditionally extracted from the parent plant roots. That is huge. It proves that by deeply understanding biosynthetic pathways and improving bioreactor design, we can make natural colorant production economically viable and completely independent of agricultural constraints. Which brings us to a final, somewhat provocative thought. We started with traditional extraction from flowers and insects, and we've arrived at optimized cellular bioreactors. As biotechnology continues to blur the line between natural agricultural growth and precisely engineered yield, what will the food colorants of tomorrow truly look like? Will natural strictly mean grown in a field or perfectly synthesized in a cell culture? I hope this explainer has provided you, whether you're a chemist, a regulator, or a food technologist, with the structural and biological insights to lead that very discussion in your own field. Thank you so much for joining me on this molecular journey.